remind the group that these are all recorded. They're on the tumor section website and also the YouTube channel for the AANS. I'd like to thank the AANS, Patricia Kaminsky, and also Maria Noor Ulhuda, who does a lot of the back door work in terms of helping organize these webinars. So, Michael, welcome, and thank you for sharing this talk with us today.
Thank you very much, Professor Michael Tucker. It's an honor to welcome you today to our webinar series. You are a very, someone sought after to speak at such an important topic. Just a little few words, just a little intro about Professor Michael Tucker. He's basically considered one of the pioneers of connectomic-based surgery. It's a very important concept that we all need to have a lot of awareness about. So Professor Sokro has served as the director of the Brain Tumor Center at the University of Oklahoma. He has penned down more than 300 papers and enjoys a very high H index, of course, obviously, for his very groundbreaking work. So it's an honor to welcome you today and to have our audience listen to you and learn more about the anatomy of the human cognitive and emotional system and how does it work, sir. Thank you very much for joining us today. The floor is all yours now.
Hi, thanks everyone. So if I had to, obviously most people know my disclosure. It crosses over with what we're talking about, but again, we're not talking specifically solely about the technology. And what I would say in this particular case is, if I had to summarize the take-home message here, and when I first put this slide up many years ago, it was a pretty good idea. Now we have pretty good evidence, and it's mounting, that if we talk about large-scale brain networks, should we make our best effort to consider them when we do brain surgery? The answer is yes. And part of the reason that got me interested in this is when you're a resident and you take out brain tumors and you're really just happy that you get through the case and nothing horrible happens. Patients can look really good in the ICU. They do this. And this has been our kind of standard for decades, proving that we didn't do something horrible. And that's across multiple domains of neurosurgery. But the reality is, if you're really thoughtful and you spend time talking to your patients and their families, even in operations that we think went well, the same person goes on and does something stupid, okay? Judgment issues are a major problem, right? And that doesn't mean we can always avoid that. If the entire right frontal lobe is a glioblastoma, you're probably not going to have someone who is high functioning at the end of it, whether or not you take the tumor out at all. What I'm getting at is that when we do it, we should have some idea what we're getting into and at least not accidentally cause this because we don't know what we're doing. So one of the issues that really started to push us in the forefront is as people got more aggressive with glioma surgery, because in short, taking a margin is always a good thing. The problem is we start running into this issue because you're taking a margin, the margin is brain, right? And again, I've been an advocate for this for a long time, to push it as far as you can. If you read my book, I am not a passive, I'm an aggressive neurosurgeon on this topic, because I think that the most patients I see who survive years out, we've done a very good surgery. It's not a guarantee that they'll do well, but it's hard to do well without it. But the reality is, you start running into these issues, and we would, if any rational actor said it, listen, given that you cannot cure this cancer with an operation, would I leave a small rind of brain if I knew it would leave the patient much better off and sacrifice probably a few cells in there when I know that there's a few cells in there anyway? The answer is any rational person would do that. But the question is, when we look at the trade-offs of causing mental health issues, of leaving patients in, yes, they can move, they can talk, but they're not really living life at its fullest. It really always comes down to the fact that there's something in this quote-unquote association cortex that seems to be important. And so we, have for a long time, when I first started talking about this 15 years ago, and people were talking about this before me, I didn't come up with this idea and conceptual basis, but there are people who said, oh, my outcomes are great. I don't need to improve on this. Well, I always raise the point, right? Are you taking out all of the tumor in all patients who walk in the door with this, and nobody has this issue, and you check? If those are four things are true, you don't need to do anything different. But there's always one of them, or usually multiple ones, that aren't true in that case. So yes, if you talk about the simplest brain tumors and you do a very cautious surgery and you don't have any bad outcomes, well, great. That's good. That's not bad. But you're really passing on a lot of things in those patients that you could probably get a better outcome if you optimize better. And ultimately, what the data shows is if some people have zero complications, then other people have 100%, because the rate of new mental health issues are 70%. So 70% seems high, and you think, wow, what does that rank among other cancers? Well, pancreatic cancer is much lower. It's about 20%. So this isn't grief in most of these people. This is that we're changing the brain. And that's fine. We don't need to beat ourselves up about it because the brain is a complicated organ. But what we need to say is that we need to continue to get better at this. And especially we need to get better if we start pushing the boundaries, right? If we're going to talk about taking margins and saying, oh, this doesn't have any consequences. Well, does it? I mean, are you living with these people? Probably not, right? And I promise you, if you start talking to people and asking, a lot of times people don't bring it up because there's other things: chemotherapy, radiation, other symptoms, hydrocephalus. There are things jumping out into the forefront of these patients. But in the long term, I promise you, if you're taking margins on a regular basis, some of these people are going to have something that will, if you ask them, you'll find out some interesting stuff. Now, I think one of the things, the unifying thing, is as we start to think about neurologic disease, we run into the same issues. Now, they're different exact issues, like whether putting a shunt in an NPH patient is a good idea or not, what a patient's going to do after stroke or TBI, almost everything about glioblastoma. And ultimately, while the question is different, the root cause of why we have more problems and have more difficult postoperative courses than orthopedic surgeons is simple. They have an organ that they understand 100%, more or less, and they can get these 97% success rates with a lot of their problems. We're dealing with a much more complicated issue, and as a result, the lack of knowledge makes us make worse decisions. And so I'm personally not wedded to any particular paradigm, but one of the things I've started to think about is that how do we really go about figuring out where function is located in the brain and avoiding it, and making sure at the very least that we're doing the least damage possible to achieve our oncologic goals. So the original approach that people took for this is wake mapping. And I'm a trained at UCSF. Obviously, I've done plenty of that. I've probably done about 1,000 awake craniotomies in my life. But the challenge really gets down to is if you even if you could find a higher cognitive quote unquote arrest site, if you don't have an idea about what you're trying to preserve, it's very hard to think that you're going to accomplish that. And the way to think about that is if you're operating the cavernous sinus and you don't know that there are any nerves running through there, the chance that you ending both removing the tumor and not having a cranial nerve deficit is very low. And so what the point we try to get to is ultimately having an anatomic target, an idea what you're here operating on, especially in situations where the anatomy is not visible to you, is critical for achieving good results. And I'm going to circle back on this several times, what we call the Big Five. And the reason we start calling this the Big Five is this is where all the evidence seems to suggest that a lot of our problems come from. There's probably other things that we don't know. We're still looking and diving into that. But these are the ones that I can say that if you're not a researchy type person, probably this is where you need to put your emphasis. And the reason I say that is at first we really ran a lot of literature. This is us screening through 20,000 papers. And of that, there are 550 different papers showing that damaging or causing problems or having dysfunction of some kind in one of these 5 networks, in the particular areas that we're showing, causes a cognitive, emotional, neurologic deficit. Since we published this paper, there have been numerous groups outside of us who've gone and found different aspects of this in a particular patient set. There's a really good paper out of the Barrow, it's in JNS if you want to read about this, about penetrating these networks. And we're not really talking about a mild cognitive deficit. We're not talking about that someone's processing speed goes down. As Duke Sampson apocryphally put it, we're not treating a cold here, guys. And I think that that's true. We can't sit there and talk about glioblastoma and worry about leaving people absolutely perfect. This isn't an olfactory group in angioma where we're trying to save smell or something. The issue in this particular case is we're not talking about that. We're talking about a drop in the Rankin score. And so these are the kind of patients who get wheeled in a wheelchair, and you don't know why they're in a wheelchair because they're not paralyzed. Those kind of people. And the bottom line is, we kind of look at this and say, well, if we have an idea what we're here to achieve, we're much more likely to achieve it. Now, let's talk about where the word network comes from. Where do we get this answer? Why do we think this is important? Well, we all kind of think about the fact that there are places in the brain that do certain things. And that's the concept of localizationism. I think almost all of us have basically looked and trained in a localizationist paradigm. And it does give us some pretty good answers, right? The precentral gyrus in almost everyone normal will have some kind of motor function in it, okay? And in most right-handed people, almost all of them, the posterior part of the IFG will have a speech function and an arrest site in it, okay? So these kind of things are pretty good, right? But the reality is it hasn't done very well in the more vague parts of the brain. And part of it is there's a lot more variability. And ultimately what we have to realize is that this was a debate that was supposedly settled 150 plus years ago when Broca and Hewlings-Jackson brought some findings that strongly pointed out to certain parts of the brain doing certain things. And they pushed out a group called the kind of more distributionist theory on this, because in part it didn't really explain the facts when we were basing on autopsy and lesion studies. And not to say that there isn't local, because we know that there is, right? But if you basically don't take a hybrid approach to realizing that there's multi-localizationism, there's multifocal functions in the brain, and that the higher cognitive functions behave like that for reasons that I'll kind of make sure, I hope I try to convey to you, then we're really going to call this vague area the quote-unquote association cortices, which is a completely meaningless term because it's not really doing that. And ultimately, we're not really going to make progress. So if you look at the top, there's 3 control networks: default mode, salient, central executive, and 2 attention networks, dorsal and ventral. Now, again, sometimes these networks do other things. These are the names they were given when they were discovered. So the default mode network isn't really, it's so familiar that it's like just acoustic neuroma. We're not going to get rid of the term in the slang, but it does different things than are really a default mode. But it shows you that, again, all of these networks are not in one place. They're distributed around the brain. And this is because they're coordinating activities across a wide area of cortex. Now, where did this concept of networks come from? Well, it really comes from people really spending time with looking at big data, particularly DTI, and more importantly, in this particular context, resting state and other types of fMRI. And when we start to look at fMRI, you're getting a signal. Okay, you're getting this activity signal, and you're getting in every voxel on the scan. So ultimately, what's really happening at the underlying function of the brain is that you have this wide-ranging set of connections that connect different parts of the brain, and it's a complex system. There are a lot of things that are redundantly or cross-wired. And again, we all look at this and say, wow, that's really beautiful, but then what does it mean, right? And we know some parts, we know the arcuate, et cetera, but the point of it is that how does this lead to function? Why does the tract, connectivity, how does it lead to personality? And that's obviously one of the great open questions in humanity. Well, ultimately, the way to think about it is that the connectome, the functional connectome, is the sum of 3 parts. One is the wiring diagram, okay? The second is that the neurotransmitters like serotonin, dopamine, and many other ones change the gains of how A, B, and C talk to each other. Then there's lots of complex waveforms at different, theta, high gamma, et cetera, that are conveyed through. And the sum of all these parts cause certain things to fire together and not fire together. And if I look at every single possible combination of that, I look at a functional connectome. The concept of networks comes from the fact that if you look at this at the whole brain level and spend time with it, there are parts of the brain that don't have identical waveforms, but they have very similar waveforms, meaning they're nearly superimposable. If one is up, the other one is up. The one that I always provide people as an example is the right and left motor cortex. They usually fire at the same time. And that doesn't mean they always do, but it means that if you statistically look at it, they will. And when, in many cases, things that we didn't even know were firing together are strongly correlated, and we call those networks. To give you kind of a visual picture of what's happening, even if you're not doing anything in particular, you're just sitting there in the MRI scanner or EEG or whatever you're using to measure MEG, different parts of the brain are firing together for short periods of time. So this is sometimes because they're talking to each other or some other more complex relationship. But the brain is cycling through all of its lower states. So when we use a task-based MRI or do a task in general, the brain function doesn't go from 0 to 60, it goes from 57 to 60. So majority of the majority of the activity is at rest, whether or not you're using the connections. And so it's part of the normal physiology of the brain. Think about it like keeping the car warm. If we begin to look at these correlations across the brain, not only can we start to learn about networks, but we can learn about dysfunction of networks. So as we started to look at the correlations at the functional connectome level, one of the things people have started to see is that people with diseases like mental illnesses and other neurologic diseases have firing patterns that don't happen in normal people. So while all of us would be slightly different if we got the same MRI every single day, there's things that shouldn't happen, and they start happening in people. And this is thought to be part of the causal pathway of mental health issues. Or what's happening when we look at those red and blue squares is that we're basically taking two areas, we're looking at their correlation pattern, and we're basically plotting a scatter plot, and then we do a correlation if there is one. Areas that tend to do common things, like motor areas, tend to be much more of a straight line. Some areas are actually opposite each other, so they're more of a negative line. And then there's things that are just random, like this. And so we would say that these two areas are not correlated, meaning they're not part of a network and they're not part of an anti-network. If we look at this at the whole brain level, and this is one of the more cited papers probably in science, it's been cited about 13,000 times. And part of the reason is people have replicated this over and over in different forms, usually within very small differences. What they did is they basically said, they took every single voxel in the brain, took its firing pattern, and clustered it with the area closest to it. And they made this map. Now, no one in the world's brain looks exactly like this. There's often jagged, intercalculated borders or enclaves and all this other thing. But what happens is that on the group level, this is almost always about what you get. So we're all variations on this at the end of the day. And so if you look, one of the interesting things that I always point out to people is that the hand motor cortex and face motor cortex live in different networks. They're closer to each other than anything else. But that kind of makes sense. It's a bilateral network in the face. The face actually has completely different connections, different fibers going into the corticospinal tract. Just because they run together at the end doesn't mean that they're necessarily doing something similar. Now, we also start to see these networks like this. So look at the brown network, and you see how it's in 4 different places in the brain. It's one at the pole, and then it goes a little bit further back. And this is the common thing of the higher scale brain networks, like default mode or language being a cognitive network. It's not the speech arrest site. It's the cognitive control of speech arrest and motor functions. So there's a cognitive component of speech, just like there's a cognitive component of motor. We call that praxis or apraxia when you lose it. And so, again, this is where the semantic language comes into the mix on top of the motoric pathways for speech. There's the dorsal attention there, the salence network. These are phylogenetically older. In general, if you think of the motor cortex and the visual system, as I go further away from that, I move up and up the hierarchy. And as I move up the hierarchy, away from the midpoints of primary cortices, it's called the cortical gradient. And ultimately, I become later in evolution, develop later in life. I use different genomics. I have different types of connectivity. So ultimately, think about this as areas being tacked on to the motor cortex to do more things. And the dorsal tension and salience network are older because they're closer to the motor cortex. Now, again, there's a lot more here, and this could be the entire lecture, but really getting to the point that's more interesting to average neurosurgeons is that the cores of these networks, the driving cores, seem to be really important for really having normal function. Most of them are bilateral, except the ventral attention network, but the reality is the more of it you lose, the less likely you are to be functioning well. So save as much as you can. It isn't quite the same as the motor cortex. It isn't always that you cut this and there's always the same problem. But in general, the more you go attack these things, the more likely you are to have a patient who's doing well. And the default mode network is probably the one that if you had to take home is canonically a pretty important part of the brain. And ultimately, it makes sense when you realize what it's doing. It's an anterior cingulate, posterior cingulate, and lateral parietal network. It's bilateral, and it does a lot of the higher cognitive functions of the brain. Now, I could spend hours talking about all the diseases that this has been implicated in. Pretty much every mental health problem. Alzheimer's disease, it's very hard to be clinically demented and have a normal DMN. It's one of the earliest things that gets hit. And we wrote a paper about this now 10 years ago in the JNS about not cutting through this system and its neighbors now. I put that in there. And this is kind of the reason why people with butterfly gliomas have worse outcomes if you don't address the anatomy, right? The salience network is also an important network. It's older. It basically is overactive in people with chronic pain, and it's misactive in people with depression. That's actually a lot of times what you're trying to do when you treat people for depression with TMS is to decouple this network from the default mode network. But also, if you look at that area of SCEF, that's actually half of the SMA. So think about this is the will, the go signal that gets people into action. So it shuts the default mode network off, which is in your own mental world, and turns on action, or vice versa. The central executive network is a network that's the difference between being a 13-year-old and a 30-year-old. Particularly on the right side, it's all the judgment issues. So, and if you look at that area 8C, that is one of the 10 most connected parts of the cortex. So it's a pretty important part of your brain. And so you want to try to keep as much of this as you can. Now, if you imagine with a frontal tumor, that's not always possible, particularly the pullover part. But again, the more of this you have, it's part of a working memory, executive function, and judgment. Now, in the interest of time, ultimately we start thinking about why is this important? And why can't we just use white matter pathways? And again, you can go back. My track record on white matter is pretty good. I've spent a lot of time talking about it. So I'm not in favor of cutting through it. I just think that it's ultimately an oversimplification of an important issue, right? So let's say I look at a bundle like the arcuate or the IFOF or something like this. Yeah, cutting through it is bad. But to say that it has a function, it's not like the median nerve, right? It's not like the radial nerve. It's not like the tibial nerve, where it has basically a small set of muscles that it controls. It's doing things that are completely diverse. So if I leave half of that intact and take the other half, it isn't that I lose half of all the functions. I lose 100% of some functions and 0% of the other ones. And ultimately, what the challenge with it is, especially as these tracks branch out to their target, if I pick them off at the target, I might as well cut the track. But more importantly, not all the fibers are equally important. If you're linking up the default mode network, you may be more important than something going to an irrelevant area. And when we start to take maps and really get to the point of what are these things actually connecting, right? And again, for those of you who've never heard me talk, I actually made this on a PowerPoint slide many years ago by hand. So I wish ChatGPT was around for that, but bottom line is I learned a lot in the process. And you can see that it's going to a lot of different areas. And that's the arc, in case anyone's concerned. So yeah, cutting through the middle of this track does devastating damage. It's necessary, but not sufficient. Because do I want to pay attention to every single one of these branches? No. I can tell you some that are probably more important than others. And one of the reasons why networks are important in doing that is that you trim the noise. You say, I need to keep this thing connected to this thing. And in order to do that, I need that fiber bundle between them. What it does is it annotates and says, this part of the track is important, this part is not. Now, in the middle of the bundle, you can't differentiate that. But if you start talking about the branches and all you have is the track branching out, you're going to put more things off limits. As soon as I put something and I say, this is doing a function, I'm making a moral judgment and then saying, you probably shouldn't take that out, or at least you should tell the patient, right? And that isn't true for every single fiber bundle, right? Every single twig of the arcuate is not equally important. And so we have to put, guideposts and say, this is important, this is not. Again, if you read this paper, we went through in extreme detail of really demonstrating what matters and what doesn't. It doesn't mean that other things don't matter or there aren't more complicated relationships that we've yet to unveil. But the short story is you're going to get better results in general the less of damage you do to these things. And also, when we start to think about problems that we understand but don't really understand, like when someone is, quote, bifrontal, we all know what that means, I hope. But ultimately, by what? Like, what part of the frontal lobe is the thing? And what starts happening is occasionally someone walks in the door with tumors on both frontal lobes. And what do you do about that, right? Especially if they're high functioning, when we're reasonably high functioning to do that. Well, it turns out it isn't bifrontal, it's by this. This is the interplay of the salience, DMN, and SMA on the medial boundary, and the frontal ascendant tracts that connect that to its lateral system are the key things that interplay between on and off. And again, if you look here, this is a paper by Mike Fox at Harvard. And I told Mike Fox about this many years ago. What he told me was, it's right here, right? And I said, how do you know? And he said, well, Red published this paper. So you look, it's very strikingly similar to that anatomy. Okay, so ultimately now we have a goal. We know what to avoid. And we started doing that several years ago. And we published this paper here. And ultimately, what we found is if you stay out of the system, you don't always not get SMA. But when you do, it's very short-lived. It's never permanent. It's mild. And oftentimes you can avoid it entirely. Now, whether you should is a good question, right? If it's epilepsy, it's different than if it's a high-grade glioma in an 80-year-old. But what I would say is it's about knowing it. If you don't know it, then you don't have the option to make that decision. It's made for you sometimes because you remove a part of the brain that does this stuff. So many people read the book. What really we're trying to do with the book is say, given what we know about network anatomy, and this is how I modified my approach when I learned this, how do we make safe cuts? If I had to remove as much of the frontal lobe as possible, what is possible, and how do I do it? You can tailor this in an infinite number of ways, but the point here is these are network respecting divisions of the brain. And so ultimately, we wanted to find how do you find these networks of the brain? So I'll zoom through this really quick. How the AI algorithm that we created works is we've trained the AI to know what a voxel of the brain really looks like from this connectivity pattern. And so once it knows that that voxel may be part of Broca's area, it can reassign it and say, well, hey, you're connected like Broca's area. And ultimately, if there's a tumor in that position, it knows if there's no tracks going to it, then it can't be Broca's area. I've got to readjust. And that turns out to be a very elegant solution. There's now been 17,000 operations done with this in the United States, and growing. It's I think every 2 hours someone does a case with this now. But basically, what the reason it's done so well is that it's quick and easy and gives a pretty good estimate of where things are when stuff's moved. This is literally the first bad case I ever put through here. This is a patient I didn't even reoperate. This came back, but my residents were so trained to get a DT on everyone that we had it. And so I said, well, let's see how it works. And we threw it in here. You can see that there's a recurrent butterflying, frontal tumor. There's been a near frontal lobectomy. If you look in the upper left panel, the insula is shifted about a centimeter and a half anterior, and yet it knows where everything is. So we're able to basically build that, put that onto the image guidance. And what we can start to think about doing is, instead of going in and shaving 1 millimeter at a time, and then we get to the really dangerous parts, slow down, which is kind of how I was taught to do this initially. What you do is you go at the dangerous parts when the image guidance is the best, and you make the cuts when the patient is able to participate, so that you don't destroy these things unknowingly. So if you think about this tumor here, you could start at the frontal pole. That's very safe, but we know where the danger is. It's in the back, it's near the midline, and now we can see it. We can see that the default mode network is smashed along the medial border. We see the central executive network draped over it. It goes up to the motor cortex. The salient network is going to be on the back of it, etc. And so we can start to think about, how do we disconnect that? Now, whether we do it as a single cut, or, conceptually, or dissect it, there's a whole bunch of ways, but it's the concept. You don't have to follow it the way I do it. I like to make a straight cut. But the reality is, even if you're doing it the way, I remember as a resident, people doing it, starting and just shaving it down. At some point, you're making this cut, right? Or otherwise, you're not taking the tumor out, whether you know you're doing it or not. So do it intentionally. You notice here, it's an aggressive surgery. It's not a GTR. We left some stuff in the basal forebrain there. We left a little bit on the DMN medially. Again, it's making it, but nobody would argue that this is a biopsy, right? This is a big surgery. And it's weighing the pros and cons of what you're doing and doing the best you can given the circumstances. Again, I didn't put the tumor in the basal forebrain, but I don't want to make the patient worse to go try to get that out. I mentioned this paper. This was Michael Lawton's paper. And again, they looked at every single cav mal that was done at the BNI over, I think it's 25 years or something. And about 9% of people rank and score dropped. Again, probably that's pretty good. But the question is, how do you get better than that, right? Well, it turns out that almost all of these patients had the same kinds of lesions. Some were things like SMA, but they're not going through the Broca's area. They're not going through the motor cortex. I mean, this is Lawton's best story. They know better than that. The bigger issue is they were going through things that we were taught were safe, like the frontal pole, or doing things near the midline, right? Turns out that's not quite the case. They don't have a hemiparesis. They just don't do well. In this case, this means a Rankin score that went down. So finally, I'm going to go into how does this arise out of anatomy? I'm going to try to go through this quick because I'm sure I'm pushing time. But ultimately, when we start to represent reality, okay, so let's say you see someone coming at you and walking towards you, and ultimately, it isn't one place in the brain where all this is experienced. It's lots of different areas simultaneously, and the sum of those parts is your representation of reality. Now, that's a state. And at a different time point, different areas can be activated, and so on. So you're representing the world as a series of states. Now, underlying this, you got all these interplays between different areas, inhibiting, stimulating, modulating. The cortex is, centers around inhibition. There's different waveforms, different neurotransmitters. But ultimately, as you go through life, you're rotating through different states. And the reason we know states are how the brain represents function are papers like this. So this is a paper out of Japan. They basically, there's about 24 core emotions. Everything else is kind of a mixture of them. And ultimately, what they're looking at here is, if you look, this is two hemispheres flattened out. The middle of the screen between those two hemispheres is the occipital cortex. So the frontal pole is on the right, the midline is at the top. And these are the regions that we've been using for years to describe brain function, the HCP regions. And you can see that different areas are active or inactive with different emotions. Now, not every area plays a role in emotion. Some areas play very little. But there isn't one emotional part of the brain. It's the sum of this. And this is argues that sort of quasi-distributional. And ultimately, we can understand this. We can look at this. And it turns out that all the emotions have a different state. So again, when we look at all the possible states in the brain, okay, It's a lot of them. There's actually 2 to 360, 363 power. And ultimately, some states just don't happen. And what's left are the states that your brain can get into. That's how you experience life. When we do brain surgery, we make that state impossible, or that state impossible. And ultimately, that's what we do when we do a surgery. We make it impossible for your brain to get in certain states. Now, it turns out that this actually helps explain eloquence, right? So we've known for a couple of years now that when we talk about areas that are eloquent, it's actually that a lot of things talk to them, and areas that are less eloquent have fewer things talking. It's called centrality. And ultimately, the reason that things like the motor cortex are inviolable is that they play a role in a huge number of states. So there's no redundant way to re-represent that. And it isn't that the state becomes a little bit less nuanced, it becomes impossible. And that's what eloquence means. So it ultimately, I think the reason it's such a good theory is that it explains the things that we already see and some things that we don't understand. Now I'm going to zoom through this a little bit in the interest of time. It's very interesting. You'll have to trust me on this. But basically, how we've been going in here and trying to figure out what states link to neuropsychological symptoms. We've got really big hyperclusters, and what we can basically do is take a neurologic disease. We published probably 50 or 60 papers on this now, and say in this case is schizophrenia. And what we can do is say, if you have this symptom, how are you different than people who don't have that symptom? And the machine learning will tell us that and point us to areas. And the nice thing is, the bare minimum, if we know where it is, we can stimulate it, we can do things to it. So we can begin to understand how these really complicated symptoms arise from the circuit and network dysfunctions that we observe in here. So what we get is we get a hot list. We can actually put them onto image guidance and show them where you are. And what's fascinating is that, obviously it's great when the machine tells you something that you expect, like that hallucinations happen in the auditory cortex. But when you learn that there's a part of the insula, that area on the lower right, and that's the only time we've ever seen this show up in a machine learning model for anything, but basically these are the people who believe that the government has inserted something into their abdomen or they have cancer. That's active, and that area tends to be actually is known to be synchronized with the stomach rhythm. It's the only part of the brain that is. So where networks come out of is that as we rotate through these states, the same configurations are useful in lots and lots of states. So the default mode configuration. Again, the DMN fire areas aren't firing all the time together. They just fire more statistically than normal. And so ultimately, what's happening is that the basal ganglia and the sum of the parts, as I talked about earlier, lead to certain configurations happening often because they're used in lots and lots of states. But why does the brain look like this? It looks kind of chaotic. And it turns out, if you look at that, someone pointed that out to me at a meeting many years ago. And I thought that looked like the spinal cord, right? And we know that the neural tube forms in a common structure. And that you see these little triads. They're represented all over the brain. They're represented there. They're represented in the basal ganglia. In fact, most of the basal ganglia, if you look, is not motor. Only that, if you look at the bottom panel where that teal is, that's the motor part. A lot of it is these higher cognitive triads. So ultimately, what we started to say is, let's roll back the clock to when all mammals are about the same, the thing on the left in development. And so in order to do that, you have to take the temporal lobe, flip it upside down, you gotta do some warping, but that's what we did. And ultimately what you see, if you look at the motor cortex, is that, again, as I pointed out, the more distant you are, you're going up a hierarchy. And these triads run along that neural tube axis repeatedly. They're hanging out and they're coordinating these really complicated behaviors. So if you look, we have 4 triads that just kind of align along that angle, right? If you look on the left, it's a lot more obvious when someone does that for you. So you imagine my excitement when we saw that for the first time. We then went to every single animal that had a cytoarchitecture map. We mapped it out. We made an estimate that turned out to be pretty true, that 2 areas with same cytoarchitecture in the same place are the same network. And people put macaques into scanners and found that to be true. and they actually have 2 fewer triads than we. We have 2. They have about 1, 2.5, probably really 2, and cats have basically none. That's the difference. So if you think that these networks aren't important, understand that you're actually taking a human and moving them back up the phylogenetic tree functionally by removing these things without paying attention to them. Now, again, they start to explain this underlying anatomy. So, for example, if you wonder why how the cerebellum looks like it does, it's actually tracing triad number 2, which is present in the macaques. But actually, most of what the cerebellum is that map. So again, these triads, what they're doing is they're hanging out in lots of different parts of the brain, and they're coordinating these complicated states to align to different functions. So yeah, the brain represents reality in multiple places at the same time, but how you can leverage that are these triads. And so ultimately, when we start thinking about why does the brain spend so much time having these big range connections, and so many of them are going to these triads and these networks, so many of these side fibers, ultimately realize that the big difference between mammals and everybody else, all the other species, is that we have these inner layers of cortex. Okay, they've got 4 layers, we've got 2. Those are the 2 layers. And if you look at what those 2 layers are doing, the outer layers are local processing. They're doing lots of parallel processing. But ultimately, the middle connections are doing long range cortical. There's also the corticospinal tract in layer 5. Layer 6 talks to the thalamus. The thalamus helps also coordinate these higher complicated functions. And so what we're really doing, and the reason archicortex came to replace a lot of these other cortex is most likely that it's coordinating long range, connectivity that we needed to really leverage our evolutionary benefit of having so much brain, and why we need to give birth live and all this other stuff that the other animals just can't do. They don't need these functions, whereas for us it's almost our whole brain. And that's why we need these big range connections. So finally, if you start thinking about what the hippocampus is doing, right, it's taking this series of states and it's putting them into different contexts. It then fires it around to the mammillary bodies, that distributes it to the cingulate gyrus. And what the cingulate gyrus is doing in all this, and this is the default mode again, is it's forcing the brain to rehearse these in sequence to put together episodic memory. Because there isn't, a memory has to be, emotional responses and visual opinion, and those are different parts of the brain, right? So how would you remember that in one place? And that's the issue, is the memory is distributed, and how it distributes itself is through that system. That's why the mammillary body has such a critical function, and we can't make new memories without it. And so ultimately, when we start thinking about how in the biology and all the different sum of the parts and different people's wiring changing over time because of the genomics, ultimately the reason we get to these abnormal states is that the gains are off, the rhythm's off, a lot of things that we don't totally understand right now, but that they lead to areas that just can't fire the way you need them to fire, or they fire ways that you don't want them to fire, and we get symptoms from that. Finally, the reason this kind of stuff is important, and the reason we've got interest in cognition, is not only to avoid destroying cognitive areas, but the idea that you can actually take action and change them. This is now getting to be an old paper. We've done hundreds and hundreds of cases since then in different parts of the world. The results we find are pretty compelling. So these are people who've had brain surgery and have a neurological deficit, speech, motor, or alertness is also in the paper. And ultimately, what we find is that not everyone with hemiparesis had an injury to the corticospinal tract or a stroke. If you stimulate the brain with TMS and you've cut the corticospinal tract, it's safe to say that this isn't going to put that back in there. What it will do is that a lot of people who have these deficits do not have injury to the corticospinal tract. What they have is you remove something that has this really complicated downstream effect of connectivity, and what happens is that the normal interactivity of the motor network, it doesn't function as a network anymore. And that's actually fairly easy to fix in some people. Sometimes you can fix this within days. And again, if you don't believe me, Sandra Krieg has published, now I believe there's the second RCT that's come out or will come out, but basically showing that the number needed to treat to improve hemiparesis in these patients is 2. So that's about one of the most effective therapies we know. And anyone who's taking care of these patients knows that even if the patients would have reached this high plateau, this good outcome, getting there faster matters. It means no DVTs, meaning that they can go into chemotherapy quicker, et cetera, et cetera. And what Sandro's data shows is that they don't reach that point, right? This is a long-term benefit. And that's what we've seen too. So I'll show you this case here. Many people have probably seen this. I think I like this case because it's simple and understandable. You can see that had an operation performed, not doing well afterwards. Now, again, this is how the workflow goes. The coil stimulates the brain magnetically through the head. And ultimately, what we need to do is make sure that it's basically over the correct location. So the question is, what is the correct location? Well, CST is intact. And you can see in this situation, correlation is shown in red. So blue is not correlated. And if you're paying attention, you see that that's a seed in the motor cortex. And the motor system works okay. There's some overconnectivity there, but look at the SMA, that blue area. That's not supposed to be like that. So that was our target. We just put that onto an image guidance screen and stimulated it. And this is what happened. Okay. That's after 1 day. So he was already up and walking. He was not walking at all. He couldn't even move the leg. And that's him after 5. So he went surfing, like, I think a week later. And so the bottom line with it is patients usually don't recover in 5 days from complete hemiparesis to that. Maybe you get lucky, but the answer is it's better if you can take action, because at the very least, you're giving them the best chance you can. And when we talk about oncofunctional balance, there are situations where you cannot save the network, or shouldn't. You shouldn't go, leave 98% of the tumor in there to save one region. It's going to disappear anyway. That's not the point. The point is that you, A, don't do it on accident. B, you don't remove a whole network to remove 1% more of the tumor. And more importantly, that you start thinking ahead of time of how, if you get a deficit, how would you reverse it? Because it's possible in a lot of these cases, if you leave good functional tissue behind, that you can get it to do what you need it to do. And more importantly, when you look, this guy didn't need to be paralyzed, obviously, right? So ultimately, what I hope I got across is that by having better information about how the brain is working and being able to think through problems that used to be intractable, or that we'd have to make really vague guesses, well, it's probably swelling. No one knows.